This is Mrs. Vance, and today's lesson is about the cell cycle. Now, as cells grow, they go through a series of events in their life. And eventually they reach the point where they need to divide. There are a number of reasons that cells might divide. For one thing, if you're a single-celled organism, like a bacterium or an amoeba, the only way you have to reproduce is to undergo cell division. So one-celled organism will divide to make two, and those two will each divide to make four, and so forth. That's the way their population numbers increase. Another reason cells might need to divide is for growth. If you're a multicellular organism like we are, you have to increase the number of cells in order to grow in size. You started out as one cell that was a union of two cells that each had half the number of chromosomes, half from your mom, half from your dad. And when those two fused together to make the one cell, the zygote, the only way you increased in size was to increase the number of cells. So by the time they were born, you had billions of cells, and now you have trillions of cells. So you have increased greatly in size and in complexity based on increasing the number of cells that you have. Another reason that cells might need to divide is for repair. When you have any kind of injury, like a cut or scrape or broken bone, those cells that are damaged have to be replaced and repaired. And that's another reason that cell division needs to occur. And then finally, the reason that we're going to talk about most of all is the cell size. The important thing that there's a critical size that cells must stay below in order to maintain homeostasis in the cell. They've got to maintain a certain surface area to volume ratio to make sure that they can take care of all the needs of the interior of the cell. Another problem can be DNA overload. If you have too big of a cell, the DNA that's in the cell can't keep up with the needs of the cell, and so the cell needs to divide to keep smaller. So what does that have to do with cell size have to do with cells dividing? Well, as cells increase in size, their volume increases faster than their surface area, or I should say their volume increases at a faster rate than the surface area. The needs of the cell for nutrients and oxygen and the production of waste is related to the volume, the size that's inside the cell contents. But the cell's ability to handle those needs, whether it's bringing in nutrients and oxygen or taking out waste, depends on the cell membrane, because all materials enter and leave the cell through the cell membrane. So the big question is, can enough oxygen and food get in and enough waste get out? Another problem with cell size is there enough DNA to support cell activities? Once the cell gets too big, past a certain size, the things that need to be done in the cell that are directed by the DNA, whether it's making proteins or doing whatever needs to be done, if the cell's too big, the needs for instructions from the DNA will outpace the amount of DNA that's present there. That's called DNA overload, and that can be a serious problem for a cell. If it can't take care of all the needs of the cell, then it needs to divide and be smaller. So what we're going to do is calculate the surface area and volume of 3 different cells to see how the rates change, and figure out the surface area to volume ratio to see why this is so important for cells. So here you have 3 different cells. Now we're using centimeters as just a convenient number because it's much more easier to work with centimeters than it is with micrometers. So let's say we have 3 cells. One is 1 centimeter on a side. They're cubical cells. The second one is 2 centimeters on a side, and the third one is 3 centimeters on a side. So calculate the surface area. You're going to multiply the length times the width and multiply that times 6 because you have 6 faces on the cube. So of the 1 centimeter cube, you have 6 square centimeters of surface area. Makes perfect sense. 1 times 1 for each side, and then you've got 6 sides. The volume is the length times the width times the height of the cell, and in this case it's 1 times 1 times 1, so the volume is 1 cubic centimeter, and the ratio of surface area to volume is 6 to 1. Now on the 2 centimeter cube, we have to calculate that also. Length times width is 4 times 6 is going to be 24 square centimeters, and the volume is going to be 2 times 2 times 2, which is 8 cubic centimeters. 24 to 8 reduces to 3 to 1. So already we've cut in half the surface area to volume ratio just by doubling the size of the cell. When we triple the size of the cell, or the length of a side, 3 times 3 times 6 is going to be 54 square centimeters for surface area. 3 times 3 times 3 is going to be 27 cubic centimeters of volume. And so the surface area to volume ratio in this case is 2 to 1. So you can see it doesn't take a real long time to get to the point where you've really decreased the surface area to volume ratio. When that happens, of course, that makes it difficult. It makes it, for one thing, it takes longer to get from the surface to the center of the cell. And for another thing, you've got a lot of stuff in between here, and there's just not enough surface area to take care of the needs of the cell. It's kind of like when we go to the pep rally in the gym and we're all trying to get out at the same time. What if we only had one door that we were all trying to get out of at the same time? It would be very difficult for that to happen. We have to have some extra exits, and that's what happens with cells when they get too big. So the cell cycle is the life cycle of a cell, from the formation of the cell till it's ready to divide to the next size. When the cell is going about its normal everyday activities, we call that interphase. Inter means between, and interphase is the time between cell divisions. The whole cell cycle, the life of the cell, we divide into 4 phases. They're called the G1, the S, the G2, and the M. G1 stands for gap 1. S stands for synthesis, G2 stands for gap 2, and the M stands for mitosis. And we're going to talk about each of these phases in turn. So this kind of shows you the life cycle of a cell. Here we've got a brand new cell, and it starts going through the first part of the interphase part, the gap 1, the G1. In the G1 phase, the cell is growing, and it's producing all the proteins it needs to make, and it's increasing the number of organelles and so forth, taking care of everything that needs to be done. At a certain point, it reaches a kind of a critical phase where it's like, wait a minute, don't quite have or pretty soon, if I keep growing at this rate, I'm not going to have enough surface area, so I need to start thinking, getting ready to divide. So then it kicks into the S phase of the cell cycle. In the synthesis phase, the DNA is being replicated. Now, DNA is a very large, complex molecule, and it takes a little bit of time to replicate the DNA. So the synthesis phase is a fairly long time in the life of the cell. Once all the DNA is copied, and so that the daughter cells can each have their own copy of the DNA, and it's verified and checked to make sure everything is right, then we go into the G2 or gap 2 phase. In this phase, we're getting ready. All the other things that are needed for cell division are being prepared. All the molecules that are needed, all the extra organelles like extra mitochondria for energy are being replicated. All of those things that are getting the final preparations for cell division. Once all that's ready, then we start mitosis, which is the division of the cell nucleus, followed by the cytokinesis, which is the division of the cell contents. And then we end up with 2 cells, each of which goes into its own cell cycle. So let's write down the information about each of the phases and what happens in those phases. In G1, the cell is increasing in size. It's got synthesis of proteins and organelles going on. The amount of time spent in this phase varies. There are some cells that stay in this phase for years at a time. If they're not an actively growing cell, like a nerve cell usually isn't, once you're grown, then we would call that, rather than a G1, we would call it a G0, where it's just kind of a steady state, not really growing, just doing everything that needs to be done. Other cells go through this pretty quickly. Bacterial cells, for instance, don't really go through a cell cycle like this because they don't have a nucleus and chromosomes, but they do go through a period of growth, and the growth and division in their life cycle only lasts about 20 minutes most of the time. Most human cells that are actively growing have a life cycle of about a day or so. Some have less, some have more, just depending on what kind of cell it is. And that's what I mean by the time varies here. The second phase is the S phase, the synthesis. This is where the chromosomes are being replicated. And again, the purpose for this is so that each daughter cell can get its own copy of the DNA, and it has everything that it needs in there to function as that particular kind of cell. Then we have the G2 phase. In the G2 phase, we have the production of organelles, especially mitochondria, because it takes a lot of energy to undergo cell division, and all other molecules needed for cell division, the components of microtubules, the enzymes that are needed to catalyze the various kinds of reactions. Everything that's needed is being prepared. And then finally, the M phase, mitosis, which is division of the nuclear contents, the chromosomes, and cytokinesis, which isn't really a phase, it's just the end of the M phase, the division of the cytoplasm that starts 2 new cell cycles for the daughter cells. This concludes the lesson about the cell cycle. The next lesson will be about mitosis.